Hey everyone, we're dissecting sheep eyes today, and the first thing that you're going to want to do is remove all of the extra material that you'll see around that eyeball. Inside the orbital cavity, you're going to have a lot of padding, a lot of fatty material that's cushioning the eye, as well as the muscles of the eyeball attached to the sclera. These are the rectus muscles. So start by cleaning that all off. You can keep the muscles or not, but we want to make sure to see the optic nerve out the back, and then have a really clear view of the sclera and the cornea in the front. So the white part of the eye is the sclera that encompasses most of that outer surface of the eyeball. And then the clear surface over your iris and pupil, this whole structure here, is the cornea, that thick layer that protects the front of the eye. These two layers make up the fibrous tunic of the eye. So fibrous tunic made up of the sclera plus the cornea. Now we'll want you to cut the eyeball in half to start to see some of the inner layers. This is best done pointed away from you. There is some ocular pressure in here, so there's fluid inside the eyeball, and you want to just take care with how you're cutting it so you don't spray yourself in the eyeball. So aim your eyeball downwards towards your pan and cut a little slit. There we go. Once you're inside the eyeball, you'll see watery liquid coming out. That watery liquid is the aqueous humor, and you're going to find a more gelatinous fluid called the vitreous humor, or the vitreous body. So we have our little puncture into the eyeball. Facing, we want to separate essentially the half of the eyeball with the cornea and the iris and the pupil, separate it from the back of the eyeball, which has the optic nerve. So just cut around the circumference and pull them apart. Looking in the back of the eye, we can see the dark inner layer. That dark layer is the choroid, and it covers that back half of the eye, as well as composing the ciliary body, which we'll take a look at in the front of the eye. Covering over that dark choroid layer, in the back of the eye you'll see a few structures. This thinner membranous structure that we're going to actually peel away gently. We can't keep it intact, really. It kind of turns into mush. But this innermost layer of your eyeball is your nervous tunic, the retina. This is a series of nerves that spreads out across the whole back of the eye to pick up sensory information for vision. So this retina is essentially a little glob now, but it was that layer, the nervous tunic, that covered the inside of the eye. As you are gently removing that retina, you may notice a place. If you look directly opposite where the optic nerve enters the eyeball, right here, on the inner part, this is your optic disc, this place where the retina is kind of really solidly attached to the back. This is where the fibers, the nerve fibers, are all gathering together to go back into the optic nerve. And it is the blind spot of your eye because there's no sensory information happening there. That is essentially a place in your eye which you cannot see. Here we can see actually coming off of the surface, the choroid is detaching a little bit. And this beautiful blue-green iridescent structure right here across the dorsal half of the eyeball, of the back of the eye is your tapetum lucidum. And this is a reflective surface in some animals that helps to send light back through those sensory structures and improve night vision. So if you see photographs of animals in the nighttime and their eyes are glowing, what you're seeing is that tapetum lucidum reflectivity. We have that whole back half of the eye where we can see our 3 layers of the eyeball: the nervous tunic, the retina, the vascular tunic, which is composed of that tapetum lucidum, and the darker choroid, that choroid that enwraps the entire inner surface of that vitreous body chamber, and the sclera, which you can see on the outside, that fibrous tunic of the eye. In this cavity, we have usually a jelly-like substance, a very gelatinous, vitreous body. A vitreous liquid is a thick liquid, so that's what you're seeing here. That eyeball jelly is filling up this chamber of the vitreous body, or the vitreous humor. Attached to it, in this case, this seems to be a little bit fused to the vitreous body. Sometimes the lens comes out with the vitreous body, sometimes it stays in the front of the eye where it came from. We can see the lens of the eye, this little pearl-like structure that has about the consistency of a Skittle. And this, if we just gently move the vitreous body away, this dark material we can see are remnants of the ciliary body, which we'll take a look at in the front of the eye. But this lens, by changing shape, can change our focal length. So if we're looking at something close up, it's one shape. If we're looking at something far away, the lens muscles in the eyeball change that lens and stretch it to be a different shape to get a better focal point for distance versus near. I'm just going to put vitreous body there, our retina in all this little pile here. There we go. And our lens. So the front of the eye has a few things to look at. This lens was sitting right here, and that circle of radiating tissue here that you can see, this little, it looks like spokes in a bicycle wheel, that is your ciliary body, and each line here is a ciliary process. So ciliary body, ciliary process, made up of ciliary processes, which are those radiating spokes. That lens would ordinarily be attached via zonule fibers, which are really, really thin, very, very thin fibers that would hold the lens in place and are part of what helped change its shape and constrict it. Though, because we removed the lens, those zonule fibers are no longer visible. And even if it was in place, they're very hard to see. But we can see we have our chamber of the vitreous body, this larger chamber in the back here. But then if we look through the iris, there's a compartment in between this ciliary body and the space where the eyeball, where the iris and the pupil and the cornea are sitting. You can see the iris through this cloudy cornea. The pupil is that opening that you can see from the back as well. Now ruminants, grazing animals, will have this horizontal pupil, and that is part of, gives them a wide field of vision to scan for predators. So if an animal is grazing, it can scan a very wide horizon with a horizontal pupil like this. Fun fact, as a grazing animal raises its head or lowers its head, the angle of its head is going to change, but the eyeball rotates so that that pupil always stays horizontal. In between the ciliary body, which you can see here, and the cornea, there's a space. That space is divided by the iris sitting here. In front of the iris, between the iris and the cornea, is the anterior chamber. So the iris divides that from the posterior chamber, which is between the iris and the ciliary body. Both of those chambers are filled with aqueous humor, and that helps to keep the eye pressurized from the outside. And that's what you saw, that more watery fluid that came out of the eye was the aqueous humor, the watery humor, versus the vitreous humor, the thicker, jelly-like humor in the back of the eye. If you look at this, how this particular eye was cut, you can see a little bit of that retina still on the surface here. So that yellowish retina, where it meets the ciliary body, the retina stops at the edge of the ciliary body. That is your ora serrata. So just to review, we have our layers of the eye, the fibrous tunic, composed of the outer layers, the sclera and the cornea, and then our inner layer is composed of the choroid, all of that, the ciliary body, in here. The inner layer of the eye is the vascular tunic, which is composed of the choroid on the back half of the eye, the ciliary body on the front half of the eye, and then also the tapetum lucidum, which is on the dorsal back half of the eye. Lastly, we have our retina, the nervous tunic, which we have removed in this case except for a few remnants, so the retina. We will also be talking about the macula, but we'll do that with an image of a living eye, so you can see light passing through that area where you can see a denser spot to identify, because it's something you can't see from a gross dissection. From this sagittal cross-section of an eye, you can actually see the lens sitting in place within the ciliary body, and you can see the chamber of the vitreous body back here. This vitreous body right now is covered by the retina, and you can see the anterior chamber here in front of the iris, and the posterior chamber behind the iris but in front of the lens. Around there you can see the sclera, the cornea, both layers of that fibrous tunic, and within that darker choroid layer, the ciliary body, and behind here the tapetum lucidum. We won't disturb that too much. Part of that vascular tunic, and then the retina, which is still sitting on top of the vitreous body. It's retinous material. Something you cannot see from a dissection that we'll be talking about in class is the macula. So that's a part of the retina that has highly densely packed nerve fibers. So it's kind of the center of vision. But we'll be able to see that looking at an optomap from an eye doctor. Thanks for listening. See you in lab. Talk to you later.